So the last part of chapter 12 is going to be about the cell cycle. So we talked a little bit about the cell cycle in class, and there's three checkpoints. And at these checkpoints, that's where the cell is going to assess certain things to see if it's okay to proceed to the next phase. So the first checkpoint is called the G1S checkpoint, and that's the most important one because that's going to be where the cell is going to look at external, internal cues and decide whether it is time to divide or not. So things like nutritional state would be a huge one in determining whether the cell is in a good enough state to go through with cell division. So what's going to happen is if it is in a good enough state, it's going to produce a protein called cyclin, and that's going to allow it to proceed to the next phase, which we'll talk about. The next checkpoint is the G2M checkpoint, and what's happened in between the G1S and the G2M, so here's the G1S checkpoint, and here's the G2M checkpoint. The big thing that's happened in between is going to be the S phase, and remember that meant synthesizing new DNA. So obviously it wants to make sure that it has copied the DNA perfectly. So what it's going to do is it is going to make sure that all of the correct nucleotides are there, and if they are, it's going to produce enough cyclin to move on to the next checkpoint. Now in here it says M checkpoint, but it's actually called the spindle checkpoint, and the spindle checkpoint is going to happen at the end of metaphase. Now let's look here and see what's happening at the end of metaphase. So we've got all of our chromosomes or sister chromatids lined up, and we've got microtubules on either side here. The spindle checkpoint is going to make sure that each side of the chromosomes are attached to microtubules, and the reason for that is because if you look at the next phase, they're going to get split. And let's look at these orange ones and say that this microtubule wasn't attached to this chromosome. Then what's going to happen is you're going to have an extra chromosome come over to this side. And that would obviously cause big problems because we talked about having too many or too few chromosomes is not a viable way to be. So that last checkpoint is making sure that the chromosomes are attached to the spindle, which are microtubules, and make sure that they're aligned correctly so that when they split, they're splitting correctly. So those are the three checkpoints. So there's going to be a molecular mechanism to this. There's not some little guy with a little, check sheet, checking all this stuff off. So phosphorylation is going to be what allows this to happen, and that's the addition of a phosphate group, and that's kind of what triggers the cycle to go further. It's kind of like the on switch. Now, you're going to have things called kinases, and ASE means that it's an enzyme, and kinases, their job is to add a phosphate. Phosphatases are in the opposite, and what their job is they're an enzyme that removes phosphates. So those start and stop the cell cycle throughout. So there's going to be a kinase, and its name is CDK, and that stands for cyclin-dependent kinase. And so what that means is that this is going to be an enzyme that depends on cyclin in order to proceed and do its job. So it's going to have to bind with cyclin in order to pass the checkpoint, is the way you can think of it. Now, there's a special type of CDK, you're going to love all these letters and numbers, and that is called CDC2. CDC2 is specifically for mammals, and that is just a special type of CDK. So what CDC2 is doing at each of these checkpoints is waiting to click in with cyclin. And if there's enough cyclin produced at that checkpoint, then it'll click in and allow the cell to go past that checkpoint. If there's not enough cyclin, then it is going to go back and retry over and over and over again. Okay, so at each of these checkpoints, there's going to be a specific name for the CDC2 connected to the cyclin at that point. So at the G1S checkpoint, if you remember, that's looking at mainly the nutritional state and size of the cell to make sure that it's healthy enough to go through cell division. If it is, you're going to have an S phase specific cyclin, and all that means is a cyclin that's produced right at the beginning of the S phase, which is the next phase, combined with CDC2. So if we want to go past this checkpoint, you have to have CDC2 bind with that S phase cyclin. That's all you need to remember for it. All right. The next checkpoint is that G2M checkpoint. And if you remember, that was the one that was looking to see if the S phase had gone correctly, which means the DNA was copied correctly. And so if it is, it's going to produce MPF, and that stands for M phase or mitosis phase promoting factor. And so that is going to be produced if all the DNA was copied correctly. Now, the last one is called the spindle checkpoint, and their signal is called APC, and that's called anaphase promoting complex. And so the whole point of APC is to say, yep, everything's okay at the end of metaphase, like we talked about here, right? Everything's okay here. So if you think about the next phase as anaphase, anaphase promoting complex is going to allow it to proceed from metaphase to anaphase. Now, what's happening here is if we were to look closely at these chromosomes, these sister chromatids together, in between them we had that little disc of protein that's holding those two together, like the two red ones and the two blue ones together. And there's actually, oops, sorry, there is actually an enzyme. What did I do with this? Oh, I lost the slide. There's an enzyme that's sitting there that is waiting to dissolve that protein holding the two red ones together. But obviously, you don't want that to dissolve the protein here or in this phase either, right? Because we want to keep sister chromatids together. So the only time we want that enzyme to be released to dissolve that protein would be here at the beginning of anaphase so that these can split apart. So if we look back at the notes, APC is going to remove inhibitors of that enzyme, and then that will allow that enzyme to separate the sister chromatids by breaking apart that protein that's holding them together. Okay, so that's what APC is. So what we're going to finish up with is just talking about the different signals that can start and stop the cell cycle. So there's a couple of different things that will make a cell cycle kind of stall out. And that is right here. So one of them is going to be anchorage dependence. And if you see in this picture, what that means is these cells are not going to do anything further if they're not anchored to something. So you don't want cells just dividing out of control, floating around your body. And so that's what anchorage dependence has to do. They have to be attached to something else before they can actually split. The next one is called density-dependent inhibition. And that's basically where you've got the cells growing, and once they reach a certain density, they stop. And you can see it tested here where they've removed a couple of cells from this dish, and then they'll just grow enough to fill in, and then that's it, they stop. Now, cancer cells are going to do something different. What cancer cells are going to do is they don't listen to any of these signals, and they just grow out of control. And that's all cancer really is just rapidly dividing cells. So the way that cancer can do that is they trick the cell cycle, and they can do that in a couple of ways. First thing is they can make their own growth factors to trick the cell into thinking that it's in an okay position to do that. The other thing is that they can make fake growth factors, so they can make something that mimics the real growth factors, or they just might have an abnormal cell cycle control system, which means they might make extra cyclin even though there shouldn't be cyclin present, or something like that. So when we talk about cancer cells, if you have cells dividing out of control, what they're going to do is they're going to form a tumor. And there's two types. There's a benign tumor, and then there's a malignant one. Now, a benign tumor is the one when you find a tumor, you're hoping that's what it is, and that's just a lump that's formed when abnormal cells remain at the original site. So you've got something that's growing, but it's just staying right there. A malignant tumor is going to be one that starts to invade the surrounding tissues, and what's even more dangerous is when they do what's called metastasizing, and that's when it exports those cells to other parts of the body where they can grow there. So we don't want a malignant tumor to metastasize because that means the cancer is spreading, basically. And so all of this cancer stuff is just basically messing with that cell cycle that we talked about. So there's many times when your cells might have cancerous precursors, but your cell cycle checkpoints are there to make sure that it doesn't go any further. So that is chapter 12, all about cell division.